Okay, I'm gonna have something here. Okay, three, two, one, go. Hi everyone, thank you for joining our biochemistry webinar. Today we will be talking about UV damage and CPD photolysis. We will explore how UV light damages DNA and CPD photolysis enzymes repair it. My name is Layla.
My name's Farhan.
And my name is Mikyas.
So, to give everyone a little bit of an introduction, life on planet Earth has been around for many, many, many years. And one of the things life has always had to contend with is our sun. So, when you were younger, your parents probably told you, hey, don't stay out in the sun for too long and put on some sunscreen. And the reason for that is because not only does the sun emit intense heat, it also emits harmful UV radiation. And this radiation could damage your DNA and do things which we do not want. And Layla will explain what that is.
Sunlight contains UVA and UVB that can damage DNA. The UV energy triggers these chemical changes in nucleotide bases, especially in pyrimidines, so base pairs like C and T, and this damage happens fast and is unavoidable in sun-exposed tissues. So this leads us to ask ourselves, how do these cells survive UV exposure when the sun is unavoidable and they're in it every day? So there are two main types of these photolesions, one being CPDs, that stands for cyclobutene pyrimidine primers, and 6,4 photoproducts, which stands for pyrimidine 6,4 pyrimidone adducts. Out of these, CPDs are most common, but they both distort the helix and interfere with normal base pairing, which then disrupts transcription. So, two ways to fix this DNA damage includes one being NER, which stands for nucleotide excision repair, which is when the damaged strand of DNA gets cut out and the undamaged strand is used as a template strand and then resynthesized DNA. The other one is photoreactivation, which is a direct reversal of CPD using photolysis and light. For a quick comparison and understanding, NER is multi-step, covers broad damage types, and works in the dark. Photoreactivation is super specific, fast, and light dependent. We chose to focus on photoreactivation solution because we're curious as to how it only needs one enzyme and a little bit of light to fix the DNA.
CPD photolase is our enzyme which is found in E. coli. It is encoded in the gene THRB and it is a class 1 CPD photolase. And the amino acid length is about 472. We looked into a database, UniProt, to find that this protein binds to two important cofactors, one being the catalytic chromatophore FAD, and the other being the light harvesting antenna MTHF. And these two cofactors are important within the activity of the catalysis, as MTHF absorbs the UV light and transfers to the FAD. And one important thing is that the FAD has to be reduced completely for the FAD, which is part of the reduced part, which has to perform the enzymatic catalysis. And here we have an image to show how the photolase works. Here, the purple and green are the DNA dimers, and our enzyme CPD photolase is the red section. And the way it works is it binds to the DNA to repair the green sections, which are Farhan, if you could point on it. The dimers around here are the CPD, which are close to the active sites where the FAD does its process of the enzymatic activity to flip it back into its proper position.
And so we used a bunch of different bioinformatic tools and websites to predict the enzyme's properties, location, and function before looking at any experimental data. So for this one specifically in E. coli, we know that E. coli is a prokaryote, so the DNA is within the cytosol. There's no nucleus. So we predicted that it would have a hydrophobic core like any other protein, and the proteins on the active site would be have some sort of hydrogen bonding. And that is exactly what we saw. So we looked at Uniprot and it told us some of the information that we knew, such as that it repairs UV-induced CPD dimers via light-dependent monomerization. And it also showed us that it has a FAD cofactor with the antenna MTHF, which we knew previously thanks to Mikyas. And it's also part of the class 1 family. Then we used PFAM and interpro domain analysis, and that basically showed us that it had two notable domains, which was the N-terminal photolyase domain and a C-terminal FAD binding domain. So these were the two major players, and also we ran it through XPath-Y, and we saw some of the basic information, such as the length, which was 472 amino acids, the molecular weight, which was 53.7 kDa, and the theoretical PI, which was 7, so completely neutral. And we also looked at some different things, such as if it had a signaling peptide within it or some sort of proteolytic peptide, which should be cleaved, but it does not. We also looked at the secondary structures and we analyzed it and we saw that it has a bunch of alpha helices and beta strands, which is to be expected, of course. And overall, without looking at any of the papers, it makes a lot of sense. It makes sense that the binding domain, right, the active site would have a bunch of hydrogen amino acids, which are capable of hydrogen bonding. And the pH and everything makes sense as well. And it works at an optimal temperature of 20 Celsius, which is specific to the organism, but all different photolysis would be specific to their organism.
We blasted the amino acid sequence to find if there's a similar type of protein, enzyme proteins. And what we found is a similar type version that is found in humans, which is the cryptochromes. There's two types, the CRY1 and CRY2. They are from the same family as CPD phosphorylase, uh, but they are different in their function. The similarities between the two is that there's a core fold that happens between the amino acid of the secondary structure of the alpha and beta, and also there's an FAD binding features that happens. The differences between the two is that the cryptochromes are not able to repair DNA. And the active site is also different as they have a criterion regulation, which is able to detect light while sleeping and be able to kind of detect light, but it does not repair DNA.
And that's to be expected since we know that evolutionarily we lost that ability. So we looked at the active site and saw that there was a lot of differences in the amino acids. And of course, that lines up with the function because as we know, structure and function usually go hand in hand.
And the primary source that we used for our webinar is the crystal structure of photolase bound to CPD-like DNA lesion. The big question was that how does CPD phosphorylase recognize a CPD and position it for repair? And why they did that with the structure of the CPD containing like the DNA duplex, and also why is this important to find how the DNA distortion and also how the lesion is able to repair it during the enzymatic activity. And Farhan would be explaining more.
And so let's look at the methods now. What they wanted to do is that they wanted to build the complex to be able to take that super specific snapshot. And so they combined the DNA, damaged DNA, CPD, with the enzyme itself. And then they use X-ray crystallography to isolate the enzyme DNA complex structure. And so this allowed them to get a very specific snapshot and be able to look at all the different angles and things like that. And the data collection conditions were synchrotron X-rays at 100 Kelvin, which is very, very cold. So what was the result? So as you can see, we see that the DNA in the complex gets bent by about 50 degrees near the lesion site, right? And so the CPD is then flipped outside of the DNA helix and it gets flipped into the active site with the FAD. And then the FAD gets split apart by the catalytic activity of the FAD. And so the interpretation is that this captures a repair-related structural substate where the repaired bases have not yet flipped back into the DNA. So what exactly is the importance? Number one, this provides a mechanistic blueprint, right? So now we kind of understand how the two work. First, you have to bend the DNA out of place, flip the lesion into the active site, place it next to the FAD, and then enable the repair chemistry. Because, as previously stated by Mikyas, the FAD has to be fully reduced. This also explains the speed of photolysis compared to NER, nucleotide excision repair. So, the structural foundation for identifying key DNA binding regions and also comparing photolysis to cryptochromes. So the bigger takeaway is humans lost photolysis, but this structure shows what light-powered DNA repair looks like at an atomic detail. And so we could look at the mechanism right here. So the FADH gets excited through light, and that's obviously with the antenna picking up the light. So then that electron is used to then interfere with the binding, and it's some complex chemistry, but it breaks it up, and then it goes back, and boom, now you got repaired DNA.
Thank you guys so much for joining our webinar. We hope you learned something new today.
Thank you, and remember to always put on some sunscreen.